Hello and welcome back to Unacademy. So let's crack NEET PG. Myself, Dr. Muskan Chaudhary, and today we'll be discussing some basic points about autoimmunity. तो आई होप कि जिन्होंने परसों 11th सितंबर को पेपर दिया एक्चुअली इट वेंट वेल सो अभी तो अपन लाइव टॉपिक्स विल कवर एंड लेटर विल स्टार्ट फ्रॉम द 18th and 19th September विल स्टार्ट विद द मेजर सेशंस फॉर द 2022 प्रिपरेशन फॉर द नीट पीजी राइट सो आज हम कुछ कुछ इंपॉर्टेंट पॉइंट्स अबाउट ऑटोइम्यूनिटी द मैकेनिज्म ऑफ ऑटोइम्यूनिटी हाउ डज इट वर्क एंड व्हाट द टर्म्स रिलेटेड टू इट एंड सम ऑफ द बेसिक डिजीज दैट वी नीड टू नो अबाउट. Well, first, starting with Unacademy, that Unacademy, as we know, is India's largest learning platform in which the topmost educators they are working day and night to provide the best of the content with the best of the T and Ds for you guys. And approx 25,000 question banks are being prepared from the topmost educators with highly effective, clinically correlated, effective explanations. And you can analyze your preparation, can prepare your basic concepts with the highly competitive quizzes, and that too all takes place within the live classes. Handler is feature that is specific to Unacademy Educator app, and it is actually something new to the education platform that Handler is this technique by the Unacademy for the online platform for the students and aspirants who are preparing for the NEET PG and all the other competitive exams. Right? Soon printed notes will be provided, but now the printed notes will be provided with the iconic subscriptions. And before 11th, it was about along with the plus it was being provided and mainly benefiting for those students who are at the peripheries and the medical officers find it quite difficult to cope up with the live sessions can actually watch the recorded sessions and whenever and wherever they get time. So in between the OPDs and if they feel like okay they can solve some MCQs they can easily do that and can watch the recorded sessions just plugging on the earphones, right? And you can study on your devices anywhere and everywhere. One is the icon subscriptions. PrepLadder has come together with the Unacademy. PrepLadder is such a great institute coaching platform for the NEET PG. The rapid revision and the snapshot of the PrepLadder are unmatchable. And video lectures from the Dream Team, along with the video lectures of the Unacademy, the TNDs of Unacademy, the question banks, and the comprehensive notes by the Unacademy, they together are being provided in the icon subscription. And your access to the best from the two of the best of the education platforms. Now, one is the 4 year subscription. So, in the 4 year subscriptions, mainly for those students who are second year, third year, and actually seek a long term commitment in the preparation for the NEET PG, they can take down the 4 year subscriptions, and it will cost you around 60,000 for 1 year, and this 4 year, and 75,000 under the iconic category, and under the plus, it will 60,000. So, when we compare this to other platforms, it is very economical and cheap, and when you divide by the for 4 years, it actually very less compared to other platforms. And where with the 12 months subscription you get 2 months totally free, and with 24 months subscriptions you get 4 months of subscription totally free, and this is a great perk. And you can use my code that is muskan10 to get 10% of the discount on your NEET PG subscription, right? So please do use Muskan 10 to get the limited time offer, and the for mainly for the first 500 students only. And you will access under the icon subscription the best of the learning platforms by the Unacademy and the Prep Ladder. And for 1 year it will cost you around 30,150, while the regular price is 4,500, and before discount it is 33,500, and after discount it is 30,000 and 150 for 1 month for 1 year. For 18 months it is 38,250, and for 2 years 42,750. For 3 years it is 51,750, and for 48 months it is all 60,750. Okay, so we'll talk about the autoimmune disease. Okay, now what is autoimmunity? ठीक है, क्या होता है ऑटो इम्यूनिटी? इट इज दैट द एबिलिटी टू रिकॉग्नाइज बिटवीन सेल्फ एंड foreign antigen. ठीक है, so what is autoimmunity? It is the ability to recognize the self and the foreign antigen that has been lost. and therefore the immunity of the person is activated against its own antigen, against its own cells, has been lost. Okay, that is ability to recognize between the self and the foreign antigen has been lost, and there are certain mechanisms by which the autoimmunity is activated, and we'll read in a little detail about that. What are these mechanisms, right? Now, what does generally happens is that whenever antigen is exposed, first a generalized reaction occurs, and then a specific reaction occurs, and in which the T cells are activated. ऐसा ही होता है, पहले एंटीजन आता है, उसको एंटीजन प्रेजेंटिंग सेल को प्रेजेंट करेगा, then APC will present these antigens to T cells, and these T cells will later activate the B cells, and these B cells will form the antibody, and they will form the interleukin 4 की help से they were activating the B cells, and other interleukins are activated. So it is what the T cells that majorly taking part in the specialized specific reactions. Now, sometimes what happens that T cells, they are for the specific antigen that has been activated. Sometimes this mechanism is lost, and the T cell is activated against all the other antigens and covering the self antigens for the self cell. So this is one of the mechanism for this, and this is called as the energy. Okay, energy is the process by which the specific reaction, this specific reaction by the T cells, they do not recognize the specific antigen. So energy, we use the term energy for it. Okay. Now, when we talk about the mechanism of autoimmunity, there are certain mechanisms that takes part in the autoimmunity, and there are different mechanisms for it. Now, first we will talk about is the loss of that losing of the energy, that is the T cells that were specialized against specific antigens. Now they are activated against all the antigens and the self antigen, and the T cells, they what? This is called as T cell energy breakdown. Okay, that means what? T cells are activated. The T cells that were supposed to be get activated against specific antigens are now activated against self antigens only. Okay, and this is seen where? This is seen in rheumatic arthritis, multiple sclerosis, or psoriasis. So this is what? This is the T cell energy breakdown, that is T cells are activated against the self antigen, and this is seen where? In the rheumatic arthritis, this multiple sclerosis and the psoriasis. Now when we talk about the second mechanism, that is a failure of, that is what? That is the failure of AICT. What is that? Is the activation induced cell death. That is a failure of AIC, that's activation induced cell death. So हम यहाँ पे किसकी बात कर रहे हैं? Activation induced cell death. किस cell death की हम बात कर रहे हैं? T cell की. कैसे? जो अगर if T cells are activated, if T cells are activated, अभी हमने पिछले वाले स्टेप में क्या देखा था कि T cells are activated against self antigens. So normally what is happens that if T cells are activated against self antigen, they will undergo lysis and apoptosis by Sjgren. So just remember when normally what does happen कि जब T cell activate हो गए खुद के एंटीजन के अगेंस्ट, खुद की सेल के अगेंस्ट, तो उनका लाइसिस या अपॉप्टोसिस को अपनी इम्यून सिस्टम कर देती है. बट दिस फेलियर ऑफ एक्टिवेशन इंड्यूस सेल डेथ ऑफ द T cells, this is again responsible for the autoimmunity. So here the cell death of T cells will not occur, and therefore these T cells that are activated against self antigen will be responsible for the autoimmunity. Right? So this is another mechanism that we talked about. One was the T cell breakdown, it is T cell energy breakdown, and the next we are talking about the failure of the AICT. Another is the loss of T regulatory cells and its functions. Okay. Now, what does the T regulatory cells do? They actually keep a check over T cells. Okay, that is they suppress the T cells, right? And they keep a check over the T cells. Now, what happens? The regulatory T cells, it is a loss. They are what? They are deactivated, and there is a loss of the function of the T regulatory cells. So, the T cells that are being activated against the self antigen, they are not checked by the regulatory T cells. So, they will again damage the self antigens and the self cells. Now, another is that the B cell with the help of the T cell. ठीक है, so as I told you कि antigen is present into the APC that will present to the T cell and that will activate the B cell. So यहाँ पे क्या हुआ कि antigen आए खुद का self antigen नहीं activate कर दिया T cell को, ठीक है, and now these T cells will activate what? Will activate the B cells and form the antibodies. This will cause the lysis and the damage of the self cells only. So this is again mechanism and it is usually seen where? It is usually seen in drug induced hemolytic anemia. That is, a patient is on any drug, ठीक है, and what does happen कि drug that will go and bind with the RBC, that is the cell bound antigen, it will become a part of the cell bound antigen कि RBC के membrane पे जाके drug will attach and a new antigen, that is RBC cell, will be modified and this will be presented to the T cells and they will think that it is not our self antigen and they will activate the B cells and form the antibodies and which will lead to a drug induced hemolytic anemia. Okay. Now next is that sequestrated antigens. Okay. That is, some antigens are hidden and where those hidden antigens are found, okay, that are found where? In the blood, in testis, in ovary, in the lens. Okay. And they have entirely different genetic material. So they have different antigens. That is not similar to the other regulatory, other normal cells of the body. So some antigens are hidden, that is normally they are not present in the blood and they are not represented to the T cells. Okay, that is present in the testis, ovaries and lens. And how they are protected? They are protected by the blood barriers. That is the one is the testicular blood barrier, that these antigens protected against the APCs, so they do not get represented to the T cells, so they are hidden antigens. But what happens sometimes is that some trauma has taken place. Okay, if some trauma has taken place, so this will expose the antigens of the testis or the ovary or the lens to the normal circulation in the blood, and this will represent to the T cells, and they will be activated against our on antigens. So this also happens in the traumatic injury of the lens. जब lens का जब traumatic injury हो जाता है, तो other lens injury also takes place automatically. You've read in the ophthalmology. So this is basic reason for the autoimmunity, क्योंकि जो antigen that was hidden for so many time and the T cells that were not activated against the self antigen, they actually get activated once the trauma has taken place for the lens. Another mechanism is molecular mimicry. And this is something that you very basically know, that certain antigens, they are shared with similar antigenic between us and the other microorganisms. And one of the classical example of it is what? The streptococcal Therefore, there is any streptococcal infection, so antibodies against that is formed against the streptococcus, the antigen similar to that of the heart. Okay, and therefore the antibodies that are formed against the streptococcus will again react against the heart cells, and this is seen where in the rheumatic fever. Is seen where in the rheumatic fever, जहाँ पे जो heart की जो cells होती है, they share the basic antigenic structure similar to that of the streptococcus, causing the molecular mimicry, and therefore this cross reaction between the antibodies that has been formed for the streptococcus, but they react with the heart cells, and it is seen where in the rheumatic fever. Now one is the bystanding feature. इसमें क्या होता है कि जनरली क्या होता है जब एंटीजन आता है, it will form an antigen antibody complex. So what happens that is this immune complex has been formed, an antigen and the antibody, they are neutralizing each other, but this antigen antibody immune complex, this will also attract the other inflammatory cells and other immune cells, and these immune cells, they start attacking getting attracted to this immune complex, will also अटैक इट्स ऑन सेल. ठीक है, सो देयरफॉर द इम्यून सिस्टम गेट हाइपर एक्टिवेटेड, जैसे एंटीजन एंटीबॉडी कॉम्प्लेक्स बना, उस एंटीजन एंटीबॉडी कॉम्प्लेक्स को देख के बाकी सेल्स है वो भी एक्टिवेट हो जाएंगी, एंड दे विल स्टार्ट अटैकिंग द सेल्फ एंटीजन्स. So this is also basic, this is very basic that I am teaching you, that is how to understand the autoimmunity. And next is the polyclonal activation. हमने पॉलीक्लोनल एक्टिवेशन ऑफ द बी सेल्स अपन कहाँ कहाँ पे देखा है. वन इज द सुपर एंटीजन, इन दैट वी हैव सीन, एंड अदर्स आर टी इंडिपेंडेंट सेल एंटीजन्स. ठीक है, जस्ट दीज आर द फॉरेन एंटीजन्स दैट आर एक्टिवेटिंग द causing the polyclonal activation of the B cells. But sometimes there is activation of the, normally what happens in the body, there is monoclonal activation of the B cells, but sometimes there is polyclonal activation of B cells that will causing self antigen damage. Now another is one is the sequestration. Although we have seen one sequestration antigen where the antigens that are hidden in the testes and the ovary and the lens, they are exposed and the T cells get activated. But sometimes what happens is that some fetal antigens that are hidden in fetal life, when they are exposed, this will lead to what? The molecular sequestration. Although it happens very rarely, but sometimes whatever these autoimmunity takes place, it is one of the, out of 10 it is the one situation that takes place, and out of these so many mechanisms, any one mechanism is responsible for the autoimmunity. Now we'll read some basics for the autoimmunity. That what is autoimmunity? Autoimmunity is nothing but a condition that is where a body's own immune cells will act against what? The self antigen, that is the immune cells of the body is unable to recognize the self antigen and the foreign antigen, resulting into the functional damage. Paul Ehrlich had first introduced the concept of the autoimmunity and termed it as horror autotoxicus. So who was it? Was the Sir Paul Ehrlich. Normally immune system doesn't act, it doesn't react with the own antigens due to the protective mechanism, that is a tolerance, and tolerance is also basically of two types. One is the central tolerance, and another is what? Another is peripheral tolerance. ठीक है, so normally the immune system doesn't react with its own cells, but sometimes it is due to the protective mechanism that is present in a body, और उसको अपन tolerance कहते हैं. और तो tolerance में दो तरह के, the one is we'll read about it later, कि तो central tolerance and the peripheral tolerance, and any breach in the tolerance mechanism will cause what? Will result in what? To the autoimmune disease. Now, therefore, before going to detail with some brief check out for the autoimmune disease, that is the immunological tolerance. Now it is mediated by the two of the mechanism, that one is the central tolerance and another is what, the peripheral tolerance. Okay, so immunological tolerance is a state in which individual is incapable of developing any immune response against its own antigen, and there are of two types. That is, one is central tolerance and another is what, the peripheral tolerance. First, we'll talk about the central tolerance. That is what this refers to the deletion of self-reactive T cell and the B lymphocytes. That is, it refers to the deletion of the self-reactive T and the B lymphocytes during their maturation in the central lymphoid organs, that is in the thymus for the T cells and the bone marrow for the B cells. So whatever the self-reactive T and B cells, what is happens the central tolerance कि उन cells का क्या हो जाएगा, deletion हो जाएगा. That is a self-reactive T cell and B cell, so that the concept of the autoimmunity is removed basically within the thymus for the T cells and the bone marrow for the B cells. In thymus, during the T cell development in thymus, if any self antigens are encountered, they are processed and presented by the thymic antigen presenting cell in association with the self MHC. Any developing T cell that express a receptor for such self-antigen is negatively selected and is deleted by the apoptosis, that we just read about AICD, that is the activation-induced cell death that is taking place, that any T cell that is activated against self-antigen is negatively selected and that is killed, and apoptosis is done by the thymus, therefore resulting in the peripheral T cell pool that is devoid of the self-reactive cells. Now, in the bone marrow, where the development for the B cell will take place while developing, immature B cells in the bone marrow encounter a self-antigen new development. Their tolerance is developed by one is the receptor editing. It is a process by which the B cell reactivate the machinery of antigen receptor gene rearrangement, so that a different B cell receptor will be produced with no longer recognize the self antigen. So even if there is some B cell that is self reactive, so receptor editing will takes place. That is, it is a genetical modification that is mainly according for the light chain. So a different B cell receptor will be produced so that it is no more able to recognize the self antigens. Next is what, the negative selection, similar to that of the thymus, that is after receptor editing the B cell again, if it again recognizes self antigen, then they are destroyed by the apoptosis. So even if it, whether it be thymus or be it the bone marrow, अगर thymus में अगर कोई भी T cell है जो selectively react against the self antigen, they are by negative selection they are taken down by apoptosis. And अगर बोन मैरो में डिपेंड करता है, पहले तो रिसेप्टर एडिटिंग से भी इट विल टेक हेल्प, कि रिसेप्टर एडिटिंग में बी सेल के रिसेप्टर्स उनको मॉडिफाई कर देंगे ताकि वो सेल्फ एंटीजन को रिकॉग्नाइज नहीं कर सके, फिर भी इट इज नॉट हेल्पिंग, देन इट विल आल्सो टेक हेल्प ऑफ द नेगेटिव सिलेक्शन and killing the cell by the apoptosis. However, the process of the central tolerance is not completely perfect. Many self-reactive T cells and B cells being receptors escape into the periphery. So, central tolerance, we talked about how does the thymus and the bone marrow with the help of receptor editing and the negative selection will take place and clear the central pool of this self-reactive T cells and B cells. Now, when we talk about the peripheral tolerance, it refers to backup mechanism that occurs in the periphery tissues to counteract those T cells and the B cells that has escaped into the peripheral pools. Now, it is provided. Now, first is the ignorance. Now, what is ignorance? It means that those self-reactive T cells that has escaped the thymus and now present in the peripheral pool, they may never encounter any self-antigen for which they have the receptors and therefore they remain in a state of what? Ignorance. तो उसको बोलेंगे ignorance. Energy that we have seen, that is the, what is it? It is defined as unresponsiveness to antigenic stimulation, that is the self-reactive T cells if they interact with the self antigen but the signal is blocked, okay, and they will be unable to activate and respond to antigenic stimulation. Normally, the T cell activation requires two signal. Main signal provided by the antigen. They will start in the initially we talked about Q1 is generalized immune reaction that is taking place, and then the specific reaction in which the T cell actually act against the self antigen. So what does happens in the A energy, that this responsiveness will occur to the antigenic stimulation by the T cell receptors, and if self antigens are processed and presented by the APCs, they do not bear the co-stimulators and a negative signal is delivered and the cell become anergic. But if this anergic background breakdown has taken place, the T cell will react with the self antigen, although it is blocked by the anergy. Now, one is what the phenotypic skewing. Okay, what is phenotypic skewing? Is a self-reactive T cell interacting with the APCs, presenting with the self antigen. They undergo the full activation but might secrete non-pathogenic cytokines. That is phenotypic skewing. That is, even if the self-antigen has acted with the T cell, they will actually secrete non-pathogenic cytokines and hence they are activated but they do not induce any type of response. Now next is what apoptosis by the AICT we have seen, that is a self-reactive T cells activated after interacted. They are presented but they induces upregulation of the fast ligand that we saw कि क्या होता है apoptosis by AICT, that is the activation induced cell death, that is the T cells that has been activated against the self antigen in the autoimmunity, they are actually called lysized, and apoptosis has been done by the regulation of the fast ligand which interact with the death receptors leading toward the apoptosis, and this mechanism is called as what? AICD. Next is the regulatory T cells, that is a track, that is they keep a check over and regulation over the self-reactive T cells through the secretion of certain cytokines, that is the interleukin 10 and transforming growth factor or killing directly. Okay, that is they keeping a check over the self-reactive T cells through the certain cytokines the 10, and so we need to remember that they does what the negative regulation of the T cells, and what are these interleukin 10 and the transforming growth factor beta. Next is what? Next is the dendritic cells. When certain dendritic cells, such as the immature and the tolerogenic, they capture the self-antigens, they down-regulate the expression of the molecules of the ligands such as CD40 and B7 molecules, or indirectly by induction of the T regulatory cells. So what is the dendritic cell is doing? Either they are down-regulating or they are inducing what the T regulatory cells. Either they are down regulating the expression of the CD40 and the B cell molecules, or they are upregulating the T regulatory cells. Next is the sequestration, that we took certain self antigens when evade immune recognition. They are actually this we talked about the testes and the ovaries, and the this lens we talked about the corneal proteins and the antigens from the brains. They are actually the hidden antigens. Now, certain self-antigens can evade immune recognition by the sequestration immunologically privileged sites, because there is blood barrier that is keeping safe from the blood, and therefore the T cells are not activated. Now, the self-reacting B cells that have escaped. Till now we talked about the T cells, but the B cells that has escaped the bone marrow to the periphery, the central tolerance, are further destroyed by spleen by several mechanisms such as down regulation of the B cell growth factor and B cell activating factor. So these are the mechanisms वन इज अ सेंट्रल टॉलरेंस जहाँ पे थाइमस था, और थाइमस में क्या हुआ कि टी सेल के डेवलपमेंट के टाइम अगर कोई टी सेल सेल्फ एंटीजन से रिएक्ट कर रही है तो नेगेटिव सिलेक्शन से उसका अपोप्टोसिस कर रहे थे। एंड इन द सेम फॉर द बोन मैरो बाय द नेगेटिव सिलेक्शन एंड द रिसेप्टर एडिटिंग, उसका जो रिसेप्टर था दैट वाज फॉर द सेल्फ एंटीजन वाज बीइंग मॉडिफाइड, एंड इफ इवन इफ द still reacting against the self antigen was the cell was lysed. Now peripheral tolerance, one is ignorance, that is it is incapable of that is ignorance means that T cell has ignored the self antigen, has never encountered with the self antigen energy, that is unresponsive to the antigenic stimulus. Next is what the phenotypic skewing, that is even the T cell has activated but is unable to induce any autoimmune response. Apoptosis by AICT, that is the T cells that has been activated are made actually removed by the activation induced cell death by the upregulation of the fast ligand through apoptosis. Next is the T check cells, that is the T regulatory cells that keep the check over the T cells, right? Next is the dendritic cells. They either upregulate the T regulatory cells or downregulate the ligands such as CD40 and B cell molecules that were taking part in activating the T cells. Another is the sequestration of the self antigens, that are certain barriers are present which are keeping the antigens protective against the T cells, and they are present where in the cornea, in the brain, in the testis. And B cells, the 10% of the B cells that are actually going to the periphery, escaping the central tolerance, they are down regulated by what? By the spleen with the help of the B cell activating factor, by the down regulation of the B cell activating factor and keeping a check over the autoimmunity. So these are some of the methods, these are the process through which the autoimmunity is prevented at every step for the T cell and the B cell. Now, coming next to the mechanism of the autoimmunity results due to the breakdown of one of the certain mechanisms that we have seen that was keeping a check over the autoimmunity, and breakdown of the T cell energy. Right, it is a breakdown of the T cell energy. Normally, cells does not usually express the co-stimulatory molecules, but can be induced to do so. Such induction may occur in the presence of the tissue necrosis and the local inflammation. And this mechanism, as I had already told you, we have seen in the rheumatic arthritis, in the psoriasis, and in the multiple sclerosis, where the T cells are activated against the self antigen, in where the T cell energy breakdown. ठीक है, where they usually do not express the B7 co-stimulating molecules, but they even do so, so they will be activated against the self antigen. So sometimes this breakdown does occurs. So this will happen in the rheumatic arthritis, in the psoriasis, and the multiple sclerosis. Next mechanism is the same, that is the loss of the T regulatory cells, and T regulatory cells was keeping a check over the T cells, and therefore protecting against the T cells, self-reactive T cells. This results in the loss of the T regulatory and causing the non-suppression of the T cells. Now, providing the T cells to stimulate the self-reactive B cells, as we know that the B cells, they are activated by the T cells to form the antibodies. So antibody response to the self-antigen will occur only when potentially self-reactive B cells receive the help from the T cells, because T cell only activate the B cell. नॉर्मली मैकेनिज्म दैट वी नो अबाउट द एंटीजन एंटीबॉडी, सो बी सेल्स जो सेल्फ रिएक्टिव है वो अकेले काम नहीं करेंगे, उनके लिए इंड्यूस करने के लिए टी सेल विल एक्टिवेट देम, एंड इफ द टी सेल्स इज एक्टिवेटेड they will activate the B cells, and self-reactive B cell will act against the on cells. For example, in the autoimmune, that is the drug induced or this autoimmune hemolytic anemia, and we saw that the drug induced hemolytic anemia that we saw, this was what the happening कि आरबीसी जो है, आरबीसी की मेम्ब्रेन मॉडिफाई हो रही क्योंकि यहाँ पे आके अपना ड्रग लग गया, तो दिस इज द सेल बाउंड एंटीजन, एंड दिस विल बी प्रेजेंटेड टू द टी सेल एज डिफरेंट एंटीजन, और ये इसके अगेंस्ट ही एंटीबॉडीज बनाना शुरू कर देगा, सो कॉजिंग व्हाट द ड्रग इंड्यूस्ड hemolytic anemia. right, causing the alteration in the red cell surface. Next is the release of the secretory antigens, and this is from the brain and the cornea and the testis that some trauma has taken place. Therefore, these antigens that were hidden for so much of time, they are now exposed to the blood and the components and to the T cells, and the T cells will not be able to recognize that these are self antigens, and they will start acting against them. The secretory antigens are usually viewed as a foreign to the immune system, as they are never been exposed to the tolerance mechanism during the development of the immune system, because they were actually hidden by the blood barriers. Injury to these organs will lead to release of such antigens, which are well capable of mounting an immune response. Okay, so when after trauma, spermatozoa and the ocular antigen, when they are released after trauma, so that is after post traumatic uveitis or the post vasectomy orchitis, what will happen, they will self T cells will start reacting against the self antigens, because till now they were actually hidden by our own immune system through the barriers. Infectious agents may even participate in the pathogenicity as well. One is the molecular mimicry that we see where, that is we see for the streptococcus for the rheumatic fever. उसमें क्या हो रहा था कि some antigens they actually the epitopes. Epitopes क्या होता है अपन ने पढ़ा था, that is the smallest subunit of the antigen that is causing what the responsible for the activating of the immune system for the antigenicity, and they actually some microorganisms they share the epitope with the human antigens, and an immune response against such microbes will actually cause the activation immune system against the self antigen, क्योंकि epitope ये अपना antigen है किसी organism का और ये अपना एंटीजन है, उसका छोटा सा एपिटोप है, वो सेम है दोनों का एपिटोप, दैट इज ऑलमोस्ट सेम. ठीक है, although or other structure of the antigen is different, but there is a similarity between the epitope, so there will be cross reactivity obviously, क्योंकि antibody बना तो इसके against इस organism का, but due to the sharing of the epitope the antigen will also react with the self cells. Okay, resulting to what? Due to what? The molecular mimicry. Due to what? Molecular mimicry. Such microorganisms, they share the antigenic determinant, that is the epitope, with the self-antigens, and immune response against such microbes would produce what? The antibodies that can cross-react with the self antigens. For example, we see in the rheumatic fever जहाँ पे क्या होता है, streptococcus organism जो है उसका जो antigen है that is actually the M protein that can react with the cardiac antigen. क्यों? Because there is a similarity between the M protein of the streptococcus with the glycoprotein of the heart cells, and therefore the antibodies that has been formed against the streptococcus will react with the heart cells, and this is seen where in the rheumatic fever. In the rheumatic fever. And this is called as what? This is called as the molecular mimicry involving the T cell epitopes, and other examples that is the multiple sclerosis where the T cell reacting to the myelin proteins. Okay, and that is why, because that is similar to that of the virus, proteins would have been induced by reacting against peptides derived from many microbes, including the virus, right? And similarly, in that of the rheumatic fevers, we see where in the multiple sclerosis, ठीक है, मल्टीपल स्क्लेरोसिस में भी क्या हो रहा है, टी सेल रिएक्ट कर रही है अगेंस्ट द माइलिन प्रोटीन्स. ठीक है, माइलिन बेसिक प्रोटीन्स, एंड दैट इज बिकॉज ऑफ द सिमिलरिटी बिटवीन द वायरस पेप्टाइड्स एंड द माइलिन प्रोटीन्स. Now next is the polyclonal lymphocyte activation. That this method also we saw when we talked about कि कैसे काम करता है autoimmunity. That is the polyclonal activation of the B cells or the T cells, right? That is several microorganisms and their products are capable of causing the polyclonal activation of the T cell and the B cells. And we read about the polyclonal activation of the B cells कहाँ पे super antigen में हो रहा है, T cell independent antigens that were causing the polyclonal activation of the B cells, causing what? Causing hypergamma ठीक है. Polyclonus T cell activation, super antigens released from the microbes from the staph and Yersinia, right in the mycoplasma, falciparum. We read so many examples, HIV, rabies, right, and that they are released from the microbes. They cause the activation of the T cell, that is a polyclonal, directly by binding to the antigenic non-specific variable beta region of T cell receptor. This was so important for the superantigens because superantigens it was due to the binding at what the variable beta region of the T cell receptors, and it can be induced. All the examples EBV, CMV, HIV, rabies, melanoma, which is the example of the superantigens, they will be included where causing the polyclonal lymphocytic activation, resulting what causing what autoimmunity. Next is the exposure of the cryptic cell epitopes, that is exposure of the cryptic cell epitopes, that research has proved that molecular sequestration of the antigen is much more common than the anatomical sequestration. Now that is what we are talking about, the molecular sequestration. That is, during development of the immune system, not all epitopes of an antigen are effectively processed and presented to the T cells. There are some non-dominant cryptic. Cryptic का मतलब ही क्या होता है? छुपा. ठीक है, cryptins में cryptic means that is something that is hidden, that is छुपा हुआ, that is non-dominant cryptic epitopes which remain sequestrated, that is some antigens that remain hidden, and hence T cell clones reacting against such epitopes are not deleted. Such cryptic self-epitopes can be released secondary to inflammation at the site of the tissue injury, which can induce increased protease production and processing of the self-epitopes by the APCs. So sometimes what happens that during trauma or this secondary to any inflammation, these cryptic epitopes that were hidden, they can be released, and they are formed during the development of the immune system, that is in the infant, that is in the infants they are present during, they are not released into the blood, and they are cryptic epitopes, but during secondary inflammation when they are released, they will be recognized by the T cell and represented to the APCs, causing the immunity, self autoimmunity. Next is the epitope spreading. Now, the self-peptides released during the persistent inflammation-induced tissue damage and are processed and presented by the APCs along with the microbial peptides. It is possible that they may occur a shift or spread to the T cell recognition to the self-epitope presented on the APCs rather than microbial epitope. So, what is happening here? The self-peptides released during the inflammation-induced tissue damage and they are processed. That is the peptide self peptides, that is a chronic any chronic infection, when the self peptides are released in the inflammation, when the self antigens are released, so they will be processed by the APCs, causing the autoimmunity. Now, one is the bystander activation, as I already told you, that is the activation of the self-reactive Th1 cells, and the or activation of the microorganism specific Th1 cell leads to cytokine influx, because as I told you, as antigen antibody complex bana, okay, so these antigen antibody complex will also attract other cells, other cytokine inflammatory cells, and this will cause the non-specific T cell activation at the site of the infection, causing what the autoimmunity. Okay. Now, some of the autoimmune disease that we'll talk about, the immunological attack of the self-reactive T lymphocyte or the autoimmune tissue leads to development of the various autoimmune disease, and their broad range which can either be localized single organ or may involve the other systemic manifestation. सबसे पहले अपन बात करेंगे autoimmune hemolytic anemia, that is easily recognized by the कोम्स टेस्ट, क्योंकि यहाँ पे ऑटोम्यून कोम्स टेस्ट अपन क्यों करते हैं, क्योंकि इनकम्प्लीट एंटीबॉडी टू डिटेक्ट द इनकम्प्लीट एंटीबॉडी, दैट इज आईजीजी एंटीबॉडी इज इनकम्प्लीट एंटीबॉडी, तो ब्लॉक तो कर देता है एंटीजन को, बट द रिएक्शन इज नॉट सीन, देयरफॉर इट इज रिकग्नाइज्ड बाय द Coombs test by addition of the anti-human antibody, in which the red cells are incubated with the AIH anti-human antibody. If IgG autoantibodies are present on the RBCs, the cells are agglutinated by the anti-serum. Next is one of the example of the autoimmune disease is the good pastures disease. That is the biopsies from the patients are stained with the anti-IgG and C3b. That is, they will show a linear deposit of the IgG and C3b on the basement membrane. That is, the self antibodies are acting against the self cells, that is IgG and the C3b. We'll read a little in detail about the SLE in the coming slides. That is, the SLE, we detect the autoantibodies, the various self autoantibodies by the indirect immunofluorescence assay and the ELISA technique. Most is the which is the most specific. We'll just read about the कौन सा most specific होता है and what the most sensitive. That is, most sensitive is ANA, and what is the most specific? That is the Smith antibody more than the double stranded DNA antibody. We'll read more about the details in the SL in the coming ugly slide. Now another lupus band test, it is can detect the deposits of the immunoglobulin. And as LE cell test that we also see in the SLE only, that is a lupus arithmetic cell test, and it was commonly used for diagnosis, but it is no longer used. Next is the scleroderma, that is the anti-sclero70 antibody is raised. Anti-SCL70 antibody is raised, detected by the same indirect immunofluorescence assay. And another is the Sjgren syndrome, and we'll read कि Sjgren syndrome में primary and secondary. Primary is glandular and the extraglandular, and in the extraglandular, what is the most commonly we see? The arthralgia and the arthritis, and the glandular we see the against the exocrine glands, that is a keratoconjunctivitis sicca and the xerostomia. Rheumatoid arthritis, that is, it is recognized by the detection of the two important antibodies, that is the RA factor and the ACPA factor. Now, RA factor by the LGT, LET, and it is an IgM. Just remember the type of the antibody is RA factor, that it is a type of the IgM that is acted against the IgG FC portion. Okay, and RA factor detection has good sensitivity. False positive is also seen in other autoimmune disease for the RA factor. But remember, what is RA factor? क्या होता है? It is the IgM antibody that is directed against the IgG FC portion. And another is the anti-citrulline peptide antibody, that is, it is also an autoantibody, and it is uh, it is more specific for that of the rheumatoid arthritis, and it is a positive in 86% of the cases with established R positive RA and 25% with the RA negative RA, but it is highly specific. That is the anti cyclic citrullinate peptide antibody, anti-CCP antibody. It is highly sensitive, more than that of the RA factor. Another is the very important test for R. For R, we do is the Rose-Wallace test to detect the RA factor. So, rheumatoid arthritis, please remember that it is the RA factor that we see. There is an IgM antibody against IgG FC portion, and another is the anti-CCP, that is the most it is highly specific, and it is anti-cyclic citrullinated peptide antibody. And the name of the test that we also read in the last classes for the antigen antibody session, that is what the Rose-Waller test. Rose-Waller test. Okay, we'll talk a little about some important few points about the SLE. Now, SLE is also an again autoimmune disease. Okay, and multiple organ disease, that is so many organs will be involved, so many system will be involved. Now, when it is lungs are involved, please remember that it is mainly cause pleuritis. And when heart will be involved, it is most commonly will cause the pericarditis. And this is very important. Remember all this. When blood is involved, this will most commonly cause the autoimmune hemolytic anemia. When kidney is involved, it will cause what? Lupus nephritis. When musculoskeletal system is involved, it will cause arthritis, but that is of non-erosive type. Okay? And it in on pathology it will show what? Periarteriolar fibrosis, and it is due to vasculitis seen where? In spleen, and will typical thinning, that is the onion skinning, is will be seen in the periarteriolar fibrosis. तो क्या याद करने की जरूरत है? It is in the lung, it will cause the pleuritis. Most commonly अगर heart है तो pericarditis, blood है तो it is the autoimmune hemolytic anemia. ठीक है, किडनी है तो इट इज द ल्यूपस नेफ्राइटिस, मस्कोस्केलेटन है तो इट इज आर्थराइटिस, right? And what is the most common system that is involved in the SLE is what? The musculoskeleton, NCM, that is the most common system that is involved is what? The musculoskeleton. And the second most system that is involved is the blood. Okay, that is the blood. Okay, now coming next is that when we talk about the lupus nephritis, okay, the type of the lesion that we typically see is what? Wire loop lesion. And they are typically seen in 345, that is 3, 4, and 5th stage of lupus nephritis, and most common in the stage 4. ठीक है, and if the wire loop lesions are present, then it will show what? The active and the poor disease, active and the poor disease. And these wire loop lesions are due to the endothelial immune complex deposition. ठीक है, it is due to what? The endothelial immune complex deposition, causing what? The wire loop lesions in the glomerulus endothelium, and causing what? The active, and it is if it is present, these wire loop lesions are present. It is mainly present stage 3, 4, and 5. It is 345, and it is a 4, that is the most common stage. Okay, and it is due to the endothelial immune complex deposition. Now when we talk about SLE, some important points, that is the most sensitive antibody, most specific antibody, best for diagnosis, for prognosis, right? What is the most sensitive? It is the anti-nuclear antibody. Most specific is what? The Smith and more than the double stranded DNA antibody. Best for the diagnosis against also dsDNA, and best for the prognosis also dsDNA. But most, what is the most sensitive? That is the ANA. Now, when we so see the immunofluorescence pattern for this antibodies deposition, the most common pattern we see is what the rim pattern or the peripheral pattern. Okay, and we see the deposition of the dsDNA. Okay, that is the rim. At the rim, we'll see the deposition of the antibodies, and therefore it is also called as the rim or the peripheral pattern, and it is the most common. So remember, most sensitive ANA, most specific Smith, best for the diagnosis for the prognosis, and the most common pattern that we see on the immunofluorescence is what the rim or the peripheral pattern, जहाँ पे अपन periphery पे deposition of the dsDNA will see. एक होता है next is what the homogeneous or the diffuse pattern we see. These are types of the pattern that we see in the IFL immunofluorescence assay. That is the homogeneous and diffuse pattern, and it is due to the deposition of the histone H4H. Homogeneous में histone proteins का deposition होगा, histone proteins का deposition होगा, and where it is seen in the drug induced lupus erythematosus. ठीक है. सो होमोजेनस में H से H, दैट इज होमोजेनस में हिस्टोन प्रोटीन्स का डिपोजिशन होगा, एंड इट इज सीन वेयर ड्यू टू ड्रग इंड्यूस्ड लूपस, एंड इट इज सीन वेयर होमोजेनस पैटर्न और डिफ्यूज पैटर्न. इसके अलावा मोस्ट कॉमन नॉन स्पेसिफिक पैटर्न that we see is what the speckled. Okay, speckle that is having the deposition of the Smith antibody, that is S speckled में Smith antibody, that is the most common non-specific pattern. इसमें क्या होगा? Dark spots, that is the background will be what? This will be green, but dark spots, that is the like the speckles. So this is what we'll see about the image. We'll also see that is the speckled pattern. एक अपन देखेंगे centromere pattern, that is green dots against dark background. We'll also see the image of this key to a dark background, but green dots will be seen. That is against the dark background, green dots will be seen, and this is nothing but the centromere pattern. And we see the small irregular deposition. Now, and where it is seen? It is seen in the scleroderma. Is seen where? Seen where? In the scleroderma. Now, next is the nuclear pattern. Okay, and they are what? They are large, regular green dots. That is, they are large, regular green dots, and seen where? It is also seen in the scleroderma and Sjogren's syndrome. Okay, where seen? It is seen where? In the scleroderma and the Sjogren's syndrome. Now, when we talk about the autoimmunity, we see one is the non-HLA associated, that is mainly seen with the gene PTPN22, and the gene is will show the polymorphism. Okay, and it is seen where? In the rheumatic arthritis, in the multiple sclerosis. के अलावा where it is seen Hashimoto's disease. Okay, and when we talk about these is what the non-HLA associated, that is the PTP and gene 22, and they will show the polymorphism and seen in RAMS and the Hashimoto. But some HLA associated, everyone knows about the HLA B27, that is the pair, yeah, that is the psoriasis, ankylosing spondylitis, inflammatory bowel disease, and the Ritter's syndrome. And then HLA 5 also we talk about, that we see where in the diabetes. Now, other than this, the HLA associated, this is also seen where the gene that we talk about is the NOD2 gene, that is associated with which disease? That is the Crohn's disease. And one gene, that is the FOXP3 genes, that is seen where in the IPEX syndrome. Associated with what? IPEX syndrome. That is I for what? Immune dysregulation, polyandrinopathy, enteropathy, and X for what? X-linked recessive. Okay, so this is some basic classification for the autoimmunity. And SLE, we talked a lot about the SLE. Now, some other important, that for photosensitivity, which is the main antibody that is associated, is the Rho, the SS antibody. And same for, that is the pericarditis in the SLE, is the same Rho. For the shrinking lung syndrome, for the neonatal lupus, for the neonatal lupus with heart block, okay, for them what? That is the Rho, that is the, and for the neonatal with the heart block is also the Rho antibody more than the La antibody, that is the SSAB more than that of the SSB. For the Raynaud's phenomena, what is the sequence for the Raynaud's phenomena? That is the PCR, that is what? The pallor, cyanosis, and the redness. And for here, that is the anti-U1 RNP, that is the anti-U1 ribonucleoprotein antibody. And another is the psychosis in SLE, that is the anti-ribosome B antibody. Okay, so these are some important antibodies as association with the photosensitivity, pericarditis, shrinking, neonatal lupus with the heart block, Raynaud's, PCR, this psychosis. Now, when we talk about the Sjogren's syndrome, is of basically of two टाइप. एक तो प्राइमरी होता है, एक सेकेंडरी होता है. ठीक है, प्राइमरी में भी दो होता है, दैट इज डिपेंड कि व्हाट इज, इट इज ग्लैंडुलर और इट इज व्हाट द एक्स्ट्रा ग्लैंडुलर. अगर एक्स्ट्रा ग्लैंडुलर है, दैट इज द मोस्ट कॉमन व्हाट विल सी आर्थ्रेल्जिया और आर्थराइटिस. ठीक है, दैट विल शो द एसोसिएशन विद marginal zone B cell non-Hodgkin's lymphoma. And for the glandular, that is the keratoconjunctivis sicca, that is the dry eye, and the dry mouth, that is the xerostomia, that is what? The dry mouth. That is the dry eye and the dry mouth, that is for the glandular. And for the extra glandular, most common is the arthralgia, arthritis in association with the B cell NHL. Now, how we can remember the Sjogren syndrome? Very easy. That it is how it is written. That is the Sjogren's. O के ऊपर दो बिंदी. That is the eye. We are talking about the. That is the dry eye. Ketoconjunctivitis sicca and the dry. Okay. Now, when we talk about the SLE, what are the criteria, the diagnostic criteria that are important to recognize, to actually diagnose what? To diagnose the SLE, उसके लिए कम से कम more than 4 criteria equal to or more than 4 criteria are important. Okay, that is at least one clinical and one laboratory criteria or biopsy showing what? Biopsy showing lupus nephritis with anti-nuclear antibody and anti-TSDNA. Whereas this one lab criteria should be there along with the one clinical criteria. Yeah, so these are the important clinical criteria. More than more for it as at least the one clinical and the one laboratory criteria or the biopsy that is proving the lupus nephritis with the this positive ANA and the anti-DNA. Now, clinical criteria that is the acute cutaneous lupus, chronic cutaneous, oral or the nasal ulcers with the non-scarring alopecia, arthritis, and what are the type of the arthritis? That is what the non-erosive arthritis. ठीक है, and what is the most common system that was involving is the musculoskeleton. Next is the blood, renal में lupus nephritis, neurological psychosis, hemolytic anemia, leukopenia, and the thrombocytopenia. Coming to the immunological criteria is ANA, anti-DNA, anti-SM, antiphospholipid antibody, and the low complements due to deposition of the complements and the direct Coombs test. Now this is again mnemonic, that is the SOAP brain MD, that is mnemonic for what the diagnostic criteria of the SLE, that is S for serositis, pleuritis, and the pericarditis that is mostly commonly seen in the lung is what the pleuritis and the pericarditis in the heart. O for oral ulcers, A for arthritis, that is what the non-erosive arthritis, and the photosensitive, that is the raw antibodies we were seeing in the photosensitivity. B for blood, that is the anemia, leukopenia, thrombocytopenia. R for renal proteins, that is LN, and ANA, anti-nuclear antibodies, immunological, neurological, and for malar rash and the discoid rash. This is again the diagnostic, that is the same criteria. Now, how do we manage the SLE? Now, how do we manage the SLE? That is for the mild disease, give the NSAIDs, and the corticosteroids are enough with the hydroxychloroquine, and for the severe diseases systemic steroids with the cyclophosphamide, right? And for the maintenance along with the maintenance with the corticosteroids and the azathioprine with the with mycophenolate mofetil. This is again the butterfly rash or the, you know, now now we can see the butterfly rash that is on the face, forming a rough symmetrical pattern over the cheeks and across the bridge of the nose. So again, the clinical presentation of the cutaneous manifestation of the SLE, and one pattern we saw due to vasculitis, that is the onion skinning that was seen in the spleen, and it was due to what? The periarteriolar fibrosis. This is again, which is showing what the periarteriolar fibrosis and the onion skinning that is present where, that is present in the spleen, and it is known as the onion skinning. That is SL is associated with the periarterial fibrosis in the spleen due to the vasculitis, due to what the chronic immunological changes causing what the vasculitis. Now, for the lupus nephritis, do remember the stages. That is the one, two, three, total six stages. Now, one is the minimal mesangial. Stage two is the proliferative. Stage three is the focal. Stage four is the diffuse. Stage five is the membranous, and stage six is the advanced sclerosis, lupus nephritis. Discuss and brief about more the SLE and the LN coming next lectures. Moving next, these are the wire loop. You can see as if the wire has been tangled around. So this is what wire loop loop lesion that is seen in what the lupus nephritis, and what were the stages that we're seeing the wire loop lesion, that was a 345, and that is most commonly seen in the stage 4 or agar wire loop lesions hai, that is the endothelium per immune complex ka deposition hai, and that is showing what the active and the progressive disease with the poor prognosis. And what is the most common stage? It is the stage 4 where we see the wire loop like lesions, that is the thickening of the periphery capillary loops of the glomerulus. This is what the full house pattern. Please remember the full house pattern means that is the presence of the immunoglobulins with the C3 and the C1q, also known as the full house pattern, and it is a characteristic for the lupus nephritis and very rarely seen. That is the full house pattern, that is what the immunoglobulins with the C3 with C1q. Now this is a pattern. We talked about the homogeneous pattern, and homogeneous pattern is also known as what the diffuse pattern, और ये किसमें दिया था due to deposition of the histone proteins, and you can see the homogeneous deposition of the histone proteins, histone antibodies, and the periphery if more brightened in green, you can see the rim or the periphery, that is the most common pattern, and it uses deposition of the dsDNA, and this is again, this is the homogeneous pattern. This is what the speckled pattern, you can see this is a rim pattern, and this is the nuclear pattern or the uniform pattern with the large green dots. You can clearly differentiate, seen where in the scleroderma and the Sjogren's syndrome. This is what the centromere pattern अपना क्या देखा था? We saw what the green dots against the dark background, and they are seen in the scleroderma. Nucleolar pattern is also seen, but here the uniform green dots is seen, and it is also seen in the scleroderma and the Sjogren's syndrome. And this is the cytoplasmic rhabdoid pattern. This is not so important. These are the pattern. Please remember the important pattern is the homogeneous, speckled, peripheral, and the nucleolar. Now this is what this is LE cell, that is the lupus erythematous cell. Okay, and this is nothing but this is a macrophage that has phagocytized the denatured nuclear material of another cell, and the denatured material is absorbing the dye. So this is also known as the LE cell, and the denatured material that has been taken by the macrophage is not known as the LE body. So this is LE cell in the blood smear. Now, LE cell, what is the LE cell phenomena? Now, anti-nuclear antibodies, they cannot penetrate the intact cell and the cell nucleus should be exposed to bind with the ENAs. And for the demonstration, the blood sample is traumatized to expose the nuclei. If this mask is engulfed by the neutrophil displacing the nucleus, this is called as the LE cell. And the basic difference between the LE cell and the LE body is what? The LE body is what? The denatured nuclear material that has been taken is the LE body by the macrophage, and this whole complete is known as the LE cell. Speckle pattern we have seen already. Okay, so speckle pattern, we have seen that it is the most common non-specific pattern is what a speckled pattern that is having SM Smith antibody deposition. That is the, you can see these dark spots like the specks that is present against the dark, this green background is for nothing but speckled pattern. So the types of pattern we have seen in the SLEs. Okay, so this was it about the today's session. We talked about the autoimmunity, the mechanism, the autoimmunity, about the tolerance we talked about, and in peripheral, the central tolerance. So peripheral tolerance में क्या हो रहा था? We were talking about the thymus and the bone marrow. तो थाइमस में अपना टी सेल का डेवलपमेंट होता है, बोन मैरो में बी सेल का डेवलपमेंट होता है, बट समटाइम्स द टी सेल एक्टिवेटेड अगेंस्ट द सेल्फ एंटीजन, तो उनको इनको इनको अपने नेगेटिवली दे वर सिलेक्टेड एंड दे वर बाय apoptosis they were killed. Similarly for the B cell, and also by the receptor modeling was done, so that after the even after the receptor modeling of the this receptor has been done by the genetic coding, even if they do not they still act against the self sense, the self cell, they were also by negative selection they were acted upon. Okay, so this was we talked about what this is what talked about the central tolerance. For the peripheral tolerance, we talked about the so many ways by which it was happening. One was the ignorance, that is the T cell, that is the ignorance we talked about, that is the T cell is unable to recognize the self antigens. ठीक है, इसके अलावा हमने अपने AICT देखा था, that is activation induced the cell death, where the T cells that were activated they were actually killed by the apoptosis by the fast ligand, that was by the AICD. Next we talked about the T cell energy breakdown, and next we talked about the B cell with the help of the T cell. नेक्स्ट वी टॉक्ड अबाउट द किसकी अपने बात की थी टी रेगुलेटरी सेल, जिनका फंक्शन खत्म हो गया था. एंड अपने देखा था वन वाज द मॉलिक्यूलर मिमिक्री दैट वी वर सीन इन द स्ट्रेप्टोकोकस इन द रूमेटिक फीवर. ठीक है, एंड द सीक्वेस्ट्रेशन ऑफ द एंटीजेंस, वहां पे सेल्फ एंटीजेंस दैट वर एक्चुअली not exposed to immune system due to the presence of the barrier in the testes, in the brain and the cornea, that is the sequestration of the antigen. इसके अलावा we also saw in the drug-induced hemolytic anemia, that is the modified antigens were taking place because drug was exposed to the surface of the RBC, and RBC cell surface was modified, and antibodies were acting against that. So these were some of the important mechanism of the autoimmunity. फिर हमने देखा SLE, we talked about the SLE, इंटेंस lung, the pleuritis, pericarditis, and the musculoskeletal system is the most commonly known. Second is the blood, and what was the most sensitive and the specific antibodies, and it's a periarterial fibrosis and the onion skinning that was seen in the spleen, and it was due to what? It was due to the vasculitis. All right. Lupus nephritis, we saw about the lupus nephritis, the wire loop lesion in the 345 stage, that is 3, 4, and 5, and most common in the fourth stage, and we'll show the active disease with the poor prognosis, and the active and the progressive disease, it resembled that in the lupus nephritis, right? The discoid rash and the butterfly rash that we studied about the SLE, and the Sjgren syndrome with the dry mouth and the dry eyes. All right, so this was it about the today's session, and do use my code that is Muskan10 to get 10% of the discount. And now the Unacademy is even providing the notes, the printed notes with the iconic subscription. Okay, and you when we compare the other about the other platforms with the icon subscriptions for four, it will cost you around the 675,000. That is very cheap and economical to other coaching platforms that is present right now. So tomorrow I'll be taking the special class as well, and please do use my code that is Muskan10. And for those students and those who have given the exam just now 3 days before, so I'll all the best for your coming results, and hope that everyone, those who prepared hard, will shine in the exam. And or those who are having doubtful, those who are still doubting that should be preparing or not, they can actually take a week and think about it. If you are having doubtful situation, you shall start it soon, and it's okay that you are right. Okay, you will be able to clear it down, so you can actually right now chill at home. Okay, so is there anything that you want me to cover up? Because from start soon, we'll start from the 18th, we'll start some important topics of the microbiology and microbiology sessions. We'll start covering up each and every topic. And first, this is how I teach the first. I'll just cover up the NEET PG types of MCQ at the starting of the class, and later the theoretical part will be covered according to professional examination, so that both the students can actually benefit it from it. Although my lectures are mostly in the English, but you can just let me know if there's something problem with the language. You can just let me know. I can help you study according to your convenience. Okay, so this was it for today's session. Please do like, share, and subscribe, and see you next time.